Guys, welcome nerds everywhere. Welcome to another biology video. This one is gonna be for all you guys who are doing cell structure or ultrastructure of the cell, looking at organelles and trying to make sense of it. But before we get into this, I just wanted to start with a quick thought for the day, which is that interdependence is better, or interdependence is better than independence. Okay? We spend our whole life, or a lot of our lives, or at least a lot of our lives growing up, thinking that our objective is to become independent. That's what we hear from grown-ups. That's what we might hear from teachers, from parents, things like that, we need to become independent. But once you become independent, the next stage of development is interdependence, which is understanding that you alone can achieve a little bit, but your maximum full potential might only be realized when you learn to work with other people. That might be your friends, it might be your family, it might be colleagues, it might be complete strangers. But you've heard all the quotes, and I won't find any of those cheesy quotes that ultimately say that we can achieve more as a team than we can individually. So I think this relates to what we're going to talk about with cell structure, okay? Because whether you're a year one student, and I think this is one of the first topics that you come across where really you get to appreciate, oh my God, A-levels is so much more in-depth than what I'm used to. And it can be the first kind of eye-opening topic, frightening topic that you try to get through. And I think rather than just muddling your way through and memorizing the structure and the function of the endoplasmic reticulum, I think it might be more powerful for you to be able to put everything into one or two or three coherent stories where you understand and appreciate, rather than remember, what the interconnectedness, what the collective functions of these organelles are and how they work together. That might be more valuable to you long term than simply individually remembering what each of the organelles do. Okay? So whether you're a year one student who's looking at this for the first time, or you're a year two student looking to do some revision, I think this could be quite a useful way to approach things, even with other topics. So let's begin with that in mind. So we're looking at a cell here. I have, on this side, I've just put a bit of a plant cell structure, and but over here we've got an animal cell. So we are looking at eukaryotic cells specifically today, but there's a whole lot, obviously there's a whole lot going on. And how do we put that into a story? Well, we will begin with the nucleus, as it is essentially it's the control center for the cell. But we need to see, like, how does it do that? How does it exert its control, right? So let's begin then. So what's our story going to be? Well, we're going to have a couple of different stories, okay, maybe three. But those three stories are going to tie everything together, okay? So one, we are going to think about maybe an enzyme. Okay, an enzyme that works outside of the cell. So it's an extracellular enzyme. It's an extracellular enzyme. So even though it's produced in the cell, it works outside of the cell. Okay. And many cells do that, right? And many organelles within the cell have to work together in order to achieve that one thing, all right? Other cells, such as phagocytes, so our other story is going to be that, well, maybe we're looking at a white blood cell, it's a phagocyte, right? And there's some proteins or some pathogenic stuff outside the cell which needs to be taken in, broken down, okay? So we're going to look at that context as well, because that needs a bit of group work, a bit of teamwork as well, okay? And finally, we'll hit the old plant cell as well. All right. And there is, again, there's the different context of the flagella itself, because many animal cells won't have the flagella, but we'll have a look, we'll address when the flagella is important, why it's important, which cells have it. With those contexts set out, then let's begin. All right, so if we want to produce an extracellular enzyme, where does the story begin? It begins with the nucleus, because the nucleus has all the genes to make all the different kind of proteins that an organism might need. Okay, so that's important that the nucleus then has that information, but it's only information, it's only instructions, that's only blueprints. So how do we get that turning into an actual protein which is going to be released from the cell? So there we have that. Now the DNA, because the DNA is so important, as you can see, the DNA is, if it's containing all the information to make all the proteins in a cell and all the proteins in an organism, clearly the DNA is very important. So the DNA has to be protected from the rest of the cell. So the cytoplasm will have enzymes that break things down. You've got other compartments like the lysosome, which are breaking things down, and all these are potential dangers, potential sources of damage to the DNA, to the genes. So we must protect that DNA, okay? So the first structure that or the next thing we're going to look at is the nuclear envelope. The nuclear envelope. Now, as you can see, that is a double membrane structure that surrounds the DNA, and its main job or role is to protect the information that is stored within the DNA, within the chromosomes, within the nucleus, okay? But also then, if we want to, let's say the gene for our extracellular enzyme is here, right? How does that become an enzyme? Well, we need to copy it into mRNA, okay? So instead of all of the DNA having to go out in order for us to produce this protein, it's only the genetic information that goes out in the form of an RNA molecule, messenger RNA. So we copy the set of instructions that we need just to make this particular enzyme, and that messenger RNA molecule is then going to go out of the nucleus. So in order for this message to go out of the nucleus to the part of the cell that's going to result in the production of the protein, and you know from GCSE that ribosomes are ribosomes synthesize proteins. Well, ribosomes synthesize proteins when they get the set of instructions in the form of the messenger RNA. So, but the messenger RNA is in the nucleus. So how does the messenger RNA get to the ribosomes that are outside? Well, they need to move out of the nuclear, or out through the nuclear envelope, through the nuclear pores. Okay, so I might just label this stuff as we go along, but actually might be too busy to label it. Yeah, I'm just going to point these things out actually. So first we had out through the nuclear pore. Okay, so the function of the nuclear pore is to allow molecules / information to go in and out of the nucleus. One of those pieces of information being a messenger RNA, which is obviously the instruction to the ribosome to make a particular kind of protein. Okay? All right, so there comes our messenger RNA out of the nucleus. It's the instructions to make our extracellular enzyme. So its next stop is going to be one of these ribosomes, which is on the rough endoplasmic reticulum. Okay, in fact, I am just going to label this stuff and hope that it does not get out of control. Okay, so here we have our nuclear envelope, and like one of these gaps gaps will be a nuclear pore. Okay, hopefully you can follow. Right, so then our messenger RNA comes out and then it will encounter a ribosome. So it will bind to the ribosome, it will attach to the ribosome. Okay, hope you can see that. And then on the inside of the rough endoplasmic reticulum, on the inside is where the ribosome, so ribosome, on the outside the ribosome reads the messenger RNA instructions, but on the inside of the rough endoplasmic reticulum, that's where the protein is coming together, that the ribosome is making. Okay, so I will draw the protein kind of being formed on the inside, right there. Okay, so essentially then, the rough endoplasmic reticulum, rough in appearance because it's got these ribosomes attached to it, the rough endoplasmic reticulum is where the extracellular enzyme is synthesized. So the rough endoplasmic reticulum is the site of protein synthesis. So that's important, that's the next stage. Okay? However, we do need to just backtrack a little bit because we need to think about these ribosomes. Now, ribosomes, as you can see, there are many. I mean, the ribosomes are even floating around in the cytoplasm. Okay, so ribosomes are a big part, play obviously a big part of the functional role of the cell. They're quite numerous in the cell as well. Now, the genes in the genes in the DNA that result in ribosome production, they are heavily used, right? So, and ribosomes are actually produced in the nucleus. So all these ribosomes that you can see here that are busy making the proteins for the cell, they themselves were produced in the nucleus. And the place in the DNA where all the ribosome forming genes are, that area collectively is called the nucleolus. So there's this darker staining bit, or this darker region in the nucleus, which is heavily used because it's so busy all the time making ribosomes. That particular part is called the nucleolus. So already we can see connected function, right? The nucleus produces the ribosomes. The nucleus contains the DNA for genes that we eventually need to make into proteins. And then the messenger RNA comes from those genes, and the messenger RNA and the ribosome then come together to allow the ribosome to make the protein. Yeah? So already we've got connected function. Next. Now, when a protein is made, it's kind of like a ribbon, right? But as you can see, when a protein is actually working, like its final functional form, it's kind of all folded up into a 3D shape. So where does that happen? It's actually the rough endoplasmic reticulum is where the 3D structure is formed. So that's another functional job of the rough endoplasmic reticulum. So the rough endoplasmic reticulum has two jobs. One, it's the site of protein synthesis, but two, it's also where the protein folds up into its final 3D shape. Okay, so this protein moves through the cisternae, the channels, the canals of the rough endoplasmic reticulum, and then at some point it gets to the end where a vesicle kind of forms from the membrane of the rough endoplasmic reticulum, and it kind of detaches and forms a separate vesicle. Okay, so the protein then moves through. When it gets to the end kind of balloons and detaches, forming a vesicle. Forming a vesicle that contains the protein. All right? And then that protein goes then to the next organelle, which is the Golgi apparatus, okay, or the Golgi body, you might call it. All right. Now, in the Golgi apparatus, now what we need to appreciate is that this protein, even though it's folded into its final 3D shape, it might not be functional before it's had its modifications. Okay, and any kind of final, any kind of modifications that need to be added to make that protein functional and work are added in the Golgi apparatus. So as this protein goes through, it will get those modifications. Things you might have heard of in terms of proteins, in terms of what those modifications might be. Those modifications could be things like the addition of disulfide bonds, things like the addition of carbohydrate chains, making them glycoproteins, things like that. Those modifications will be happening here in the Golgi apparatus so that the protein can become finally useful, functional, working. So we'll just, and the protein will move through the cisternae of the Golgi. As it does so, it will move through the different sacs here. So it will move through and it will end up at the outward face of the Golgi apparatus. And what happened before, a pinching off of a part of the rough endoplasmic reticulum to form the transport vesicle, is again going to happen. This time, a part of the Golgi apparatus will pinch off and form for us, this time, however, a vesicle which contains the protein called a secretory vesicle. So that's the job of a secretory vesicle. So just label it up there. Secretory vesicle. The secretory vesicle has the packaged, modified protein, and it moves it towards the cell membrane, where the vesicle then fuses with the membrane of the cell and unloads its contents essentially outside of the cell in a process called exocytosis. Okay, so I hope that we're happy with that, and I hope that we've seen, like, this is, you know, a great example of individual functions coming together to perform one overall function. And that works on many, in terms of organisms, it works on almost every level, right? Various tissues work together to form the organ, right? Various different organs come together to form organ systems, right? And then you have obviously all these different organ systems work together to give the overall organism its life, its function of living, right? And you can even move that up. It's exactly what we were talking about at the start, right? People can't live on their own. Ideally, you know, you want to form connections with other people, work together, and so on. So there we have it again, right? So let's continue that discussion then, because there's one unsung hero for all this process, which is, how did the vesicles like randomly just get to where they were supposed to be? And the answer is they didn't. They didn't get there randomly. They were moving according to directed paths, and those paths were a part of the cytoskeleton. Okay, so there's three parts of the cytoskeleton that I will mention today and that you need to know about. Okay, so cytoskeleton part 1 is the microtubules. I've drawn them in green here. Okay, so these microtubules, they are protein cables, right? Think of like thick steel cables, and that's essentially what these microtubules are for the cell. Okay, however, their function is not strength, their function is transport. It's what these microtubules do is essentially they provide tracks or tram lines or train lines through the cell, and everything that moves from one place to another in the cell, or everything that has a particular position in the cell, is directed to its position along these microtubules. So these vesicles, the transport vesicle, in order to get from the rough endoplasmic reticulum to the Golgi apparatus, that vesicle moved along a microtubule for it to eventually get to its destination. Similarly, the secretory vesicle, in order to get from the Golgi to the cell membrane, it moved along microtubules. And various other things in the cell will be moving from place A to place B along microtubules, sometimes from in to out, sometimes from out to in. Okay, so cytoskeleton, microtubules, that's their contribution to this particular function, as well as many others. In relation to microtubules, you might also have heard of the centrioles, right? Centrioles, weird little pair of structures. Okay, now they have various roles at various stages of a cell's life. You might have heard of their role in cell division, forming the spindle fibers. Well, the spindle fibers are essentially microtubules, and the centrioles, therefore, are the organizing center of all the microtubules. That's where all the microtubules originate from, right? The centrioles. So the centriole has a very important role in kind of transport and other processes at different stages of the cell's life. Okay, right then. So now that that's done, I'm just going to make sure that my labeling, I've labeled up all the key things. So we talked about centrioles. I'm just going to label the centrioles. Okay, we talked about the microtubules. Right, did we label up our transport vesicle? All right, and careful that is different from our secretory vesicle. If you look at actual images or electron micrographs of secretory vesicles, they'll be bigger, they'll have a packaging or contents which are much more dense and dark looking, right? Whereas, from these will be quite small. So we got the transport vesicles. What else? We have now talked about the rough endoplasmic reticulum, that its job was protein synthesis, and something else, the folding of the protein also. And that's that, right? So at this point, I hope we have seen an example of interdependence, collective, individual functions contributing to overall processes. Okay, so let's just recap that and remind ourselves of what that story was. DNA had the information to make our extracellular enzyme in the form of a gene. But, and because all that information is so important, it has to be protected by a double membrane, the nuclear envelope. However, information from the nucleus does need to make its way outside, because that's where the protein synthesis machinery is, that's where the ribosomes are. And information goes from the nucleus to the ribosome via the nuclear pores, these whole type structures within the nucleus that allows regulated in and out movement through the nucleus. Okay, but again, the ribosomes, they had to be formed somewhere, and in fact the nucleolus is where the ribosomes are formed. Once they're formed, they go through the nuclear pore and attach to the rough endoplasmic reticulum. Once there, the ribosome reads the messenger RNA, reads the message, reads the instructions, synthesizes the protein in the rough endoplasmic reticulum. It's then the rough endoplasmic reticulum's job to fold that protein up into the right shape, and then that protein travels via transport vesicle to the Golgi apparatus, where it's modified. Remember, it might not be ready to work yet, where it's modified, packaged into a secretory vesicle, and the secretory vesicle deposits it out of the cell via exocytosis. Okay, and all of this being coordinated along the cytoskeletal microtubule tubules, kind of just allowing everything to happen in an organized way. Okay? And that's that. We've still got some organelles that we haven't spoken about, and we're going to speak about them again. We're going to try and put it into some kind of context. Okay, so let's begin then. Now, one thing we haven't spoken about yet is the smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum is very closely related to the rough endoplasmic reticulum, as its name suggests. Smooth endoplasmic reticulum, SER. Now, I would like you to just take a second and just look at what I've drawn here. Apart from the DNA here, which I should have drawn in a different color, all the blue lines, and even this green line, all the blue lines that you can see here are all made of the same thing, which is membrane, okay? Which is lipids, which phospholipid bilayer. If that's not meaning anything to you, it eventually will, but don't worry. For now, all you need to really understand is that all the blue lines are membrane. And membrane is important because it's barrier. It keeps things out and it keeps things in, and it allows things to develop its own specialized function, right? And so the membrane is important, but where does membrane come from? The nuclear envelope, membrane. Rough endoplasmic reticulum, membrane. Golgi apparatus, membrane. Vesicles, membrane. Cell membrane, clearly membrane. So where does all this membrane come from? It doesn't just magic out of thin air. And all this membrane originates in the smooth endoplasmic reticulum, because the smooth endoplasmic reticulum's job is to synthesize lipid-based things. Lipids, cholesterol, hormones, all those things that have a very, what's the word? All those things that are fat-based, lipid-based, hydrophobic, those things are synthesized in the smooth endoplasmic reticulum. In contrast, the rough endoplasmic reticulum's job is to make protein stuff. Okay? So we've got protein, rough endoplasmic reticulum, smooth endoplasmic reticulum, lipid-based, fat-based. So, and that's where it comes from. And membrane is lipid-based. Okay? Yeah, it's oily stuff. So that's where all this membrane comes from. So all the membrane that the nuclear envelope is made of, all the membrane that the rough endoplasmic reticulum is made of, all the Golgi apparatus membrane ultimately originated from the smooth endoplasmic reticulum. Okay? So that's the smooth endoplasmic reticulum, and I hope that you can appreciate then how it connects up to everything else. It's not just something that does its own thing. Its function is critical to everything else even existing. Okay, so there we have the smooth endoplasmic reticulum. Then other things, let's move on and let's take a slightly different look here, and maybe I could even kind of draw like a little line like this, indicating that perhaps we're just looking at a slightly different type of animal cell here. So this might be like some kind of liver cell, or a pancreatic cell, whose job it is to make enzymes. But here now we're looking at a different kind of cell. Here we might be looking at a white blood cell, a phagocyte. Okay, and again, pancreatic cells don't have flagella, so even here I might draw a line like this, indicating, generally speaking, that this might be a slightly different kind of animal cell in this section, because pancreatic cells don't have flagella. Yeah, white blood cells won't be secreting extracellular enzymes and so on. Okay, so what we're saying is that this could be a sperm cell. This right here could be a white blood cell, a phagocyte, and this might represent like a more generalized cell. Okay? Now, what we're going to look at now is some other organelles which we haven't looked at yet. So what we're going to start looking at now is lysosomes. Okay, so lysosomes, their role is a bit more involved in phagocytic cells that carry out phagocytosis. Remember the process of engulfing and internalizing pathogens, bacteria, things like that, and then digesting them, breaking them down so that they are no longer harmful. So that's the story we're going to follow up top right here. So how might a cell do that? So phagocytic cells, remember what they do is they extend their membrane around something like a bacteria or a group of bacterial cells. They might extend their membrane around that. So how does a cell change its shape? Also, phagocytes, in order to go towards a site of infection, they need to be able to move. In order to be able to move from one place to another, they need to generate cell shape changes. So how does a phagocyte change its shape? A phagocyte changes its shape by using the cytoskeleton. Okay, so we've already looked at one contribution of the cytoskeleton, namely the microtubules. But now we're going to look at another part of the cytoskeleton, which is the actin cytoskeleton, or also known as the microfilaments. So these are relatively much thinner filaments, I should say, but what they do is they get polymerized, and when they start to form these long filaments, they push the membrane out. And in doing so, they cause the membrane to reach forward towards these bacteria and start to engulf them, okay? Beginning the process of phagocytosis. So that this process of pushing out the membrane is carried out by actin filaments. So we shall name them. Okay, so this is now, remember, this is cytoskeleton, cytoskeleton 2, actin filaments. Actin filaments are involved whenever cells need to change their shape, not have their shape. If a particular cell is a cuboidal cell or if it's a spherical cell, that baseline shape, that's not determined by the actin cytoskeleton. But whenever a cell wants to change its shape, it wants to extend at the front, it needs to withdraw from the back, those shape changes are brought about by the actin cytoskeleton. So this little example of the membrane extending itself around those bacteria, that's the actin cytoskeleton at play right there. Okay? Now, when the phagocytosis continues, young people. Okay, so much important stuff going on. Okay, so, and then eventually these bacteria are going to be internalized and we will get the formation of an internal vesicle. Okay, so this, I mean, we're not going into depth in the process of phagocytosis here, okay, but this is called a phagosome, it's a glorified vesicle at the end of the day. Okay, but it's going to contain these things. So once the process of engulfing finishes, we're going to have an internalized vesicle. Now we need to destroy the contents of that vesicle, and in order to do that, we are going to merge the contents of this with the contents of another important organelle, this one called the lysosome. Okay, now to lyse something means to split something, right? Lysosome. So essentially we're going to break these pathogens, these bacteria up so that they're no longer harmful, can't cause infection. So what's going to happen is that this vesicle is then going to move towards the lysosome. Yes, likely microtubules are going to be involved in that process to get the vesicle from one place to another. But when that happens, when this vesicle comes close to the lysosome, they'll come together and fuse so that the contents of the vesicle that contains the bacteria mix with the contents of the lysosome. What does the lysosome contain? It contains hydrolytic enzymes. Proteases, nucleases, carbohydrases, lipases, enzymes that can break down these bacteria. Whatever they're made of, it can break those things down, carry out hydrolysis, break them down into harmless components. Okay? But yeah, if you think about it, where did the enzymes in the lysosome come from? They were made, there was a gene for the lysosomal enzymes. The lysosomal enzymes got expressed as the mRNA. The mRNA was used to make a protein of these enzymes. Those enzymes got modified in the Golgi apparatus, and then the vesicles moved to the lysosome to deliver those enzymes to the lysosome, and now they work there. Okay, so you see how it all works together. Okay, so the lysosome then has these enzymes break things down, job done on that front. Okay, so that's a white blood cell doing its job there, and we're looking at some organelles working together in the white blood cell. Let's now look at the sperm cell, and another component of the cytoskeleton, not another one, but one we've already mentioned. If we look at a sperm cell, its kind of unique feature, among other things, is that it's got a flagella for propulsion. Okay, so again, maybe every single cell doesn't have a flagella. Maybe every single cell doesn't have as highly adapted kind of lysosomal components. Okay, but what we do have is every single cell has its own version and specialization of its basic things, right? So every single cell has a microtubule cytoskeleton, but this will be specialized and adapted in sperm cells, right? Even muscle cells have a highly adapted and specialized cytoskeleton so that it can be contractile. But its basic components are those of any other cell. Okay? So we've got the sperm cell here. It's got a flagella. Okay, so let's just label that. Flagella. Now, a flagella is basically its long microtubule filaments that push the cell membrane out to form this hair-like or tail-like structure, right? And it's essentially, its function is propulsion, okay? But even then, like, in order for the flagella to carry out its function of propulsion, it needs energy in the form of ATP. And where does that ATP come from? It comes from organelles. Well, it comes from the mitochondria. Right, it comes from ATP, and the ATP is generated by the mitochondria, okay? So the mitochondria carries out aerobic respiration. The food that is available to a cell, it's broken down into molecules such as glucose, and the mitochondria, through various intermediate steps, can make use of the glucose and produces various substances, but also as a result of that converts the energy that's stored in glucose into ATP. Okay? And producing ATP, then it provides the energy for the flagella to move in a way that can propel the cell. Okay? And while we're here at the mitochondria, it's really very, very important for us to mention that all these processes that we spoke about, from transcription, copying the information in the DNA, from translation, putting those amino acids together by the ribosome, putting those amino acids together to make a protein, this takes energy. Any process where we're making big molecules from smaller ones, it takes energy. So that needed energy, protein synthesis needed energy, making these vesicles and moving them from one place to another, that takes energy. And then finally, letting the protein escape from the cell via exocytosis, that takes energy too. So we can't forget the role of the mitochondria in all of the other functions of the cell as well. Okay, so hopefully we're all kind of understanding that it takes all of these organelles to perform the function of the cell. Okay, and yeah, and then well, not finally, but there is one other cytoskeletal component. So, well, actually, while we're here on flagella, we should also mention that some cells also have, some cells, not sperm, but some cells also have structures. Just thinking about where I can put this thing. Some cells also have kind of smaller hair-like structures called cilia. Okay, so I'll just draw cilia right there. And these cilia are, again, they can have various functions, from being sensory to moving fluid over the cells, right? But again, these cilia are formed from microtubules. Okay, so just like flagella, they're kind of like mini flagella, if you will, but they don't have the same, because they're so small, they're not really going to move the cell, but they have other functions. Sensory functions, they can have functions of moving fluid around the cell, as we've already said, but in common with the flagella, they are based on microtubule architecture. Okay, so we have that. So actin, we've talked about in terms of changing cell shape. Flagella, cilia, based on microtubules, we've talked about, connected to the centrioles. Finally, though, for the cytoskeleton, there is this other little thing called, well, there's something in the cell which kind of pervades the whole cell. It goes through the whole cell, and it's even part of the nucleus, giving the nucleus its shape. But these things, this is what we call the cytoskeleton 3, cytoskeleton 3. These are the intermediate I hope I'm still in the shot. Yeah, great. Okay, so there's something else also in the cell. It's protein based. Again, they're filamentous proteins forming these long cables that run through the cell. But this is another type of filament, and its job is not to change the cell shape. Its job is not to transport things from one place to another. Its job is simply to give the cell its basic shape and form. Okay? So, some cells you have are very tall, like in the epithelium. Some cells are very flat and spread out. Some cells are very long, like neurons. Well, whatever the inherent differences in the shapes that cells have, that inherent shape is determined by the intermediate filaments. It literally is the basic scaffold of the cell. Okay? And I think that is it. Now, what we haven't discussed is cell wall and chloroplasts in the plant cell, so we'll just quickly mention those. Now, again, chloroplasts are in plant cells, they're not in animal cells. But a plant cell will have these organelles plus the chloroplast. And the job of the chloroplast, like, how is it connected to everything else? Well, it's got a very fundamental role, because the chloroplast carries out photosynthesis. And without photosynthesis, you don't get molecules like carbohydrate, like protein, like lipid, like nucleic acids. So without the photosynthetic process, you don't have any molecules to build anything with in the first place. So the chloroplast, literally, it is important in providing the materials with which you can synthesize and produce the rest of biological matter. Okay. Now, for an animal, an animal will consume these molecules, but through the diet, but a plant will make these molecules because it doesn't eat, right? So it synthesizes these molecules from scratch, essentially. Okay, and finally we have cell wall. Again, animal cells don't have a cell wall. It is, again, that's connected to the function. The cell wall provides structure. It prevents the cells from bursting from too much water entering. Right now, because animal cells have their own water regulation mechanisms, we don't need a cell wall to stop the cells from bursting because we're regulating our water levels. Things like plants, they don't have those kind of mechanisms, so they need that cell wall there to prevent the cell from bursting and, provide support to the rest of the plant. And there you have it, guys. If nothing else, I hope that what we've got from this is that these organelles, they work together. They perform their function for other organelles, and in order for them to perform their function, they need the other organelles. Okay? Not unlike people. Okay? So, I hope that that's made some sense. I hope that this will help you in terms of understanding why there's just so much goddamn stuff in the cell. And I hope that because there's a reason for all the stuff to be there, it makes it just a little bit easier to understand why the cell is the way it is and why you have so much revision to do and so much to learn and so much to understand. Okay? Guys, it's been an honor and a pleasure. I hope I've managed to help you a little bit. Good luck in what you do, guys. See you next time.